All right, so welcome to part 3 of lecture chapter 7. This is going to talk about action potentials. So action potentials are going to be large in terms of their depolarization, and action potentials are used for neurons to communicate over very long distances. Neurons can be very, very long. So if you think about the motor neurons that are moving your toes, the body of that neuron is actually in the spinal cord. So the spinal cord, which ends a little bit of, in the lower back area, it has to go all the way down. So that's a very long neuron, and messages have to travel very quickly. So action potentials are used to do that. Neurons will have excitable membranes, and they have the ability to generate the action potentials. So it's all about the depolarization again. Action potentials require large depolarization and very rapid events used for this communication. And action potentials travel along the axons, right? We know that they are going to exit, they travel away from the cell body on the axon towards the axon terminal. If it is a sensory neuron, then the action potentials are going to come from the receptor to the terminal. Okay, the receptor one, don't worry about that. We'll talk about that in the senses unit. So let's just take a look overall at the events that happen during an action potential. So here we are at resting potential at negative 70 millivolts. The first phase is a rapid depolarization, right? So we can see that phase 1, well, actually, let's just talk about from negative 70 to negative 55, right? This is going to be that initial stimulus caused by graded potentials. So if those graded potentials sum and they reach threshold at negative 55 millivolts, then an action potential is automatic. And so we can see that the depolarization stage starts here once we reach threshold. So threshold will cause this rapid depolarization. We're reaching a positive membrane potential. Then we're going to return to normal. So we're going to go back down. Phase 2 is called repolarization. We go past threshold, we go back down to resting potential, and then we actually dip below resting potential for a little bit, and that's called the hyperpolarization region, so after hyperpolarization. And then eventually we're going to go back to resting potential, right? So let's take a look at what's going on in terms of the channels that are opening and closing. So again, let's just understand, let's actually just draw a neuron, right? So if we have our dendrites of the neuron, we have the body of the neuron, and then we have the long axon. So in this region, we have our graded potentials, mostly caused by ligand-gated ion channels. So a neurotransmitter binds, you'll have these small depolarization events, but it can sum, and if all those depolarization events happens to reach threshold at this initial segment of the axon, then the depolarization event, right, this when it reaches negative 50 to negative 55 millivolts here, at this point we have voltage gated ion channels. So the voltage that reached the axon hillock is going to open up these ion channels. So we're not considering neurotransmitters anymore. We're considering the voltage that has reached the axon hillock. So when the voltage reaches the axon hillock, we have two voltage-gated ion channels that are of importance. We have a voltage-gated sodium channel and a voltage-gated potassium channel. The thing to remember is that the sodium channels are fast, they open fast, they close fast, and then the potassium channels are slow. They are slow to open and they're slow to close. So we can see at the time 1 here that we have the depolarization wave coming. So it's already reached threshold and we're going to begin to depolarize. So the sodium channels are going to open. Okay, so they're first to open. When sodium channels open, the sodium is going to flood into the cell, right? So the positive charges are going to come into the cell, causing that membrane to become more and more positive. Here, so this is our depolarization. So the depolarization is due to the sodium channels opening. If you look at the second time, our potassium channels are still not open. But by our third picture here, we have something happening. So our potassium channels finally open here, right? So our potassium channels are opening, and potassium is going to go into the cell. Oh, I'm sorry, potassium is going to leave the cell. So the positive charges, potassium is going to leave the cell. But when the potassium channels are opening and potassium is leaving the cell, these positive charges are going to move out of the cell, causing the membrane to go more negative. The sodium channels are inactivated. So there's actually a little lid that swings to inactivate the sodium channels. So at this point, they don't work. They're not going to be coaxed open in any way. They're completely inactivated, and you can see that it does become activated again shortly after, but there is a period of time where they're inactivated and unable to allow sodium to flow through. So during the repolarization process, what's happening is the potassium channels are causing the repolarization. Okay, so let's take a look at, this is just a closer look. This is what your textbook has in terms of the activation and inactivation of the sodium channels here, right? So we have our gate is closed but capable of opening. The depolarization event comes, the gate's open, right? It's voltage gated, and so sodium can flood into the cell. Then there is an inactivation event, right? The inactivated gate closes and it is unable to allow sodium to go through again. So it's not that it's just closed and able to open, it is completely shut, deactivated, right, or inactivated. All right, so let's just take a look at what's going on with the ions here. So we have these different events here, reaching threshold, right? So when we reach threshold, we are depolarizing the cell. When we reach threshold at -5 millivolts, the sodium channels will open, and sodium is going to flood into our cells. The positive charges are coming in, and we're going to move towards equilibrium potential of sodium, right? Remember that was, I think, positive 60. So we're moving towards that equilibrium potential for sodium. However, right, they are going to inactivate, and then the potassium channels are going to open. So during this time, we're going to stop the influx of sodium, and we're going to begin the efflux of potassium. So we're going to drive the potential more and more negative. And since the potassium channels are very slow to close, they're going to have this efflux occur much longer. And remember that the equilibrium potential for potassium was -94 millivolts. So it's actually even more negative than your resting potential. So it's going to cause the hyperpolarization to dip below action potential. I'm sorry, below your resting membrane potential. And then when the when the potassium channels close, then the membrane resting potential, the sodium potassium ATPase or the sodium potassium pump will take over and start to equalize the membrane potential back to resting. And at which point you see the line here going back to resting. Okay, so we have permeability changes for sodium and potassium during an ion channel, right? So we have if we just look at the purple line, that's just the permeability for sodium, You see a rapid increase of permeability during the depolarization phase, a rapid decrease of permeability in the repolarization phase. And for potassium, right, the permeability doesn't occur until we start to depolarize. Okay, so in terms of sending an action potential, the stimulus that causes an action potential, it doesn't matter how large the stimulus is. Action potentials are created once you reach threshold at negative 55 millivolts. So you can see that if we look at the two graphs here, or the two pictures, we can see that we have a couple of sub-threshold stimuli. We can see the small depolarization events, and then we have a threshold stimulus, right? So we reach negative 55 millivolts, and there's an action potential that takes place here. We can have a bigger stimulus, a supra-threshold stimulus, but it doesn't mean that you have a greater action potential. You can see that the action potentials, right, this action potential 1 and action potential 2, they're exactly the same. And so it doesn't matter if the stimulus barely reaches threshold or if it overshoots threshold, you will still get the same action potential. So how is it that a neuron can convey a sort of small signal, right, or a great signal? Like, how can the neuron pass on the information of just the intensity of the stimulus? So let's answer some questions, and then we'll answer that question that I just posed. So what happens during depolarization in an action potential? Go ahead and read your choices. All right, so the answer is letter A. Sodium rushes in and the membrane potential becomes positive, right? This is kind of just a fun one I saw from the textbook. Why are this toxin, tetrodotoxin, I can't say it, from pufferfish, saxitoxin from dinoflagellates, and the shellfish that eat them, neurotoxic to humans? So this is a nice example of, I'm sure you've heard rumors or stories of people eating a certain part of the pufferfish and then just keeling over dead. And it happens quite quickly because the toxin works on your neurons. And if you're unable to send an action potential, then you're going to die, right? Your nervous system just shuts down. So what is happening here? Just out of fun curiosity, you can make your guess, but it's actually letter C. They block voltage-gated sodium channels. So if you block voltage-gated sodium channels, you're not able to depolarize the cell, right? The neurons are not going to be able to depolarize, and therefore you're not going to have an action potential. During the repolarization in an action potential, what happens to the membrane permeability of sodium and potassium? So what happens during the repolarization? All right, it should be letter C. Okay, the permeability of potassium increases and the permeability of sodium decreases. All right, action potentials cannot sum together. They are all or nothing. Which type of summing can graded potentials do? So graded potentials can sum spatially and temporally. Okay, so this is just a quick comparison between graded potentials and action potentials. Again, the graded potentials are located on the dendrites and cell body, and they are also in sensory receptors, so I haven't talked about that yet. Action potentials are only found at the neuron. Graded potentials can be weak, and they're proportional to the strength of the stimulus, and then they dissipate with distance, right? Action potentials are the same all the time. So when we looked at that graph, it doesn't matter how strong the stimulus is, but as long as you reach threshold, the action potential does not change in nature. So that's where the all or none phrase comes in. You either have an action potential or you don't. There's no big or small action potential. So my question was, how does the intensity of the stimulus get passed on if your action potentials are all the same? So the direction, the graded potentials can be depolarizing or hyperpolarizing, but action potentials are always just depolarizing, right? That first initial step, you have to depolarize. Summation, we know that graded potentials can be summed, and there's no summation that happens during action potentials because action potentials rely on graded potentials to reach threshold. There are no refractory periods in graded potentials, but there are 2 refractory periods in action potentials, and I haven't talked about that yet. I will go ahead and talk about that. What kind of channels are involved in producing your change in potential. So we talked about ligand gated, so this is where neurotransmitters would bind to the receptor, or mechanically gated, which I haven't talked about yet, but it's basically where there is a physical deformation of the neuron, and that physical change of the cell causes an action potential to occur. Depolarization to occur, and you get a graded potential. And then for action potentials, the channels, right, there are voltage-gated sodium channels, voltage-gated potassium channels. Ions involved, there's a few of them. I haven't talked about them in specifically yet, but for action potentials, only sodium and potassium. And graded potentials are very quick, and a few milliseconds to seconds, and so are action potentials, 1 to 2 milliseconds. Okay, so let's talk about these refractory periods. During the time that follows an action potential, there is a decreased ability for the membrane to become excitable. There's two types of refractory periods, which are called absolute and relative. So the absolute refractory period basically spans all of the depolarization and most of the repolarization phase. What this means is the neuron is creating an action potential already, and so you cannot put another action potential on that neuron. The neuron cannot create another action potential because it's basically in the midst of doing its first action potential. So refractory periods are basically this period where you cannot create another action potential. Okay, so it's in the process of depolarizing and repolarizing. During this time, the sodium gates are inactivated, and they're just not physically able to allow sodium to rush in again. You cannot create another depolarization event to make another action potential. All right, relative refractory periods are the last part of the repolarization phase, so at the tail end of the repolarization phase, and then also during hyperpolarization. And you can create a second action potential, but you need a much stronger stimulus to do it. And this is because at this point in time, just a few milliseconds after, some sodium gates are closed, but that means that they can still get opened again, while others are inactivated. So it's going to take a much stronger stimulus to open up these sodium channels that are closed. So let's just take a look at the graph here. We have an absolute refractory period where basically nothing, no stimulus can cause that neuron to create another action potential because it's in the midst of doing the action potential anyway. And then after our rapid depolarization and hyperpolarization, at the tail end of repolarization and hyperpolarization, there is this relative refractory period where it's possible to get another action potential, but the stimulus would have to be very great. Okay, so this is just putting words to what I just mentioned. Okay, so during this absolute refractory period, no stimulus, regardless of the strength, can generate a second action potential. And then here, when we're starting to repolarize, right, this first part of it, a stimulus much stronger than normal is necessary to generate a second action potential this early in this relative refractory period. And then if we go even lower, we're reaching membrane potential again. A stimulus a little stronger than normal is necessary to generate an action potential. Okay, so that should make kind of a little bit of sense to you, because at this point we're almost back at the resting membrane potential. Okay, so because we have these refractory periods where once the neuron fires an action potential, it cannot double up on an action potential. What that means is that, I like to begin with this one, it means that there is a unidirectional propagation of action potentials, and action potentials cannot go backwards. So let me show you a picture here. Let's look at this, right? So if we have, here's our axon hillock, all right? This is where we're going to have our concentration of voltage-gated ion channels. We can see we have resting potential where inside the cell is negative and outside the cell is positive. here's where the depolarization is occurring, right? So this area has reached threshold and it's beginning an action potential. So you can see the charges are flipped, right? The inside is positive now and the outside is negative. Now, it doesn't move backwards because we don't have voltage gated ion channels in the cell body. So when the voltage depolarization occurs, then it's going to cause the membrane adjacent to it, just right next door, right where site B is, that's going to get stimulated. So the voltage gated ion channels there will get stimulated to create an action potential. So if you look at letter C, letter site B is now having an action potential, right? It's depolarizing, but it doesn't travel backwards. So the depolarization in site B does not create another action potential on site A because site A is in its refractory state. So in order for site A to create another action potential, it would have to be a much, much stronger stimulus coming from this side, okay? So that refractory period means that we can only get propagation of action potentials going down in one direction. So site B is depolarizing. Site B is going to stimulate the next little membrane adjacent to that to create an action potential, right? So if we look here, now C is having a depolarization event, but B is now in its refractory state. So the action potentials cannot travel backwards to B, they can only go forwards to D, because D is ready to go, it's at resting potential. Now, if you look here, site A is not in refractory, it's in resting, and it's able to fire another action potential. So there is a period, a very small period of time that the membrane that just had an action potential cannot fire another one. And that's the reason why action potentials always go forward and not backwards, okay? Because there's always this refractory period that prevents a quick succession of another action potential. All right, so coming back here, right? So that's why we have this unidirectional propagation and action potentials don't go backwards. And then remember that we have an all or none principle. We don't have a little big action potential. We either have an action potential or we don't. And then the frequency coding, this is where I'm going to answer the question. So how, if action potentials are just all or none events, what is it that can dictate a strong stimulus versus a weak stimulus, right? How do your cells communicate, oh, this is a really strong, intense stimulus, or this is a very light, barely there stimulus? And that's the frequency coding. So the strength of the stimulus is due to the frequency of action potentials over time. So let's take a look at a picture of this. So this is our, sub-threshold stimulus. We didn't quite reach threshold. Now we've reached threshold, right? We're going to create an action potential. If we have a stronger stimulus, right? The stimulus strength is a little bit stronger, right? So it goes above its supra-threshold. We can get many action potentials happening in that same amount of time, right? So the time scale here is the same, but we're only sending one action potential with a stimulus that just barely made threshold. And then in our second example here, we have 4 action potentials created from a stronger stimulus, a stronger supra-threshold stimulus. And then if we even have a stronger stimulus, right, that's even greater, right, then we can see that we have many action potentials, right? 1, 2, 3, 4, 5, 6, 7 action potentials happening in that same amount of time. So intensity, right? How do you convey to your body, your brain, that we have a very light signal, or we have a medium signal, or we have a very big stimulus signal? It's the frequency of action potentials. So it's how many action potentials reach, are created, and that messages are going to reach the cell on the receiving end. The cell on the receiving end is going to get many, many action potentials happening all at once. And so it's going to just have a stronger signal conveyed communication to the post synaptic cell. All right, so what is the response of an action potential to a very strong stimulus? And this should be hopefully easy. We're going to increase the rate of action potentials. All right, so I talked about the just propagation of the action potentials here. So if we remember, I'm just going to erase some of these marks I made. If a neuron is unmyelinated, then the action potentials will move incrementally down the axon, right? So we talked about how one little area is going to initiate the action potential. It's going to cause the little segment right next door to then also create an action potential. So action potentials will move down the axon of an unmyelinated axon slowly, relatively slowly, because every piece of that membrane needs to become depolarized. By comparison, if there is a myelinated axon, okay, so remember your cells that myelinate. They are your Schwann cells in the peripheral nervous system, and they are the oligodendrocytes in the central nervous system. Remember that myelination occurs in little chunks, and then we have areas where we have exposed axon. Those are called nodes of Ranvier, and we can see that if action potentials started here at the axon hillock, that the depolarization event is happening at a node before, will happen, here before we have myelination. And then that depolarization will skip the entire area where we have myelin because it's insulated, and we can't actually have transport of ions through the many, many layers of myelination. So basically what happens is it jumps. You can see the arrows here, right? The depolarization event, the positive charges are actually going to move all the way to our other node and cause a depolarization event at another node. So this kind of action potential propagation jumps from node to node to node. This is called saltatory conduction. Okay. Saltar means to jump in Spanish. So this is a jumping kind of conduction system, and it's much, much faster than the unmyelinated axon, where every single piece of that membrane needs to depolarize. Here we can skip chunks and just depolarize at the nodes of Ranvier. Okay, so messages that need to be sent very, very quickly. Most of the time, the axons will be myelinated. There's all different kinds of, neuron cell types. There's a table I'll show you, but here's another factor affecting propagation is the diameter of the axon. So if you have a very large diameter axon, you have less resistance here, and it moves much faster than a smaller diameter axon. You have more resistance, and the propagation of the action potentials are slower. And then, of course, we have myelination, right? Saltatory conduction, when you have myelination, it's much, much faster. It's about 100 times faster than if we don't have a myelination. Okay, so which of the following results in the fastest conduction velocity? You should see here a fat or large diameter axon with myelin. So just taking a look at, there's no need to memorize anything here, but just a nice example of how we have different fiber types. And remember the word fiber, a nerve fiber is the axon. Okay, so a nerve fiber, there's different morphologies, different anatomies of these long axons. Some of them are myelinated, these are not, C fibers are not. You can see that they have different functions, right? And then the diameter here, right? So they have different diameters and different conduction velocities. So you can see that your body really gives the most velocity, right? The fastest conduction is for skeletal muscle contraction. And that makes sense in terms of survival, because if you need to move very quickly out of harm's way, and so you want the messages to get to your skeletal muscle very quickly. And then other things are maybe not as fast. Still, everything's really fast, but relatively speaking. All right, so that is the end of part 3.